Our laws, as it pertains to substances, are draconian and bizarre. The psychopaths start this way. He was an alcoholic because of social media and pornography, PTSD, love addiction, fentanyl and heroin. Ridiculous. I'm a doctor, for sake. Where the hell do you think I learned that? I'm just saying, you go to treatment before you kill people. I am a clinician. I observe things about these chemicals. Let's just deal with what's real. We used to get these calls on Loveline all the time. Educate adolescents and to prevent and to treat. If you have trouble you can't stop and you want to help stop it, I can help. I got a lot to say. I got a lot more to say.
Welcome, everybody. I do plan to start a little Clubhouse a little bit later, but right now I'm watching the restream. As we always point out, we are live today on Rumble, Twitch, YouTube, Twitter, Facebook, and leave anything out, anybody? All the platforms that are available.
Why later? Why aren't you starting it up? Because people like to hear the conversation and know what we're talking about.
What happens is people pile in and then they pile out.
Well, let them pile in and tell them we're going to talk for a little while and then we'll be back to them.
Okay, as you wish.
I know, honey. I'm sorry, but I'm the boss.
Susan decided that she and I should be
mixing
you up a little more on the air. And so here we go. So I am starting a Clubhouse room, and we will allow those people to come up once they pile in.
And just explain who's here.
In a minute, I'll be introducing my guest, but right now I'm talking about what we're doing here. After I interview the guest, we may take some questions for him, and afterwards I will take a little bit of questions for me. The guest is Dr. Peter Chin Hong, Associate Dean for Regional Campuses, Medical Director specializing in ID, infectious diseases, particularly diseases in immunocompromised patients. He directs the immunocompromised host infectious disease program at UCSF. He has research focusing on donor-derived infections in transplant recipients, as well as molecular diagnostics of infectious diseases in those with suppressed immune systems. Dr. Chin Hong, welcome to the program.
Thanks so much for having me on, Dr. Drew.
Am I pronouncing your name correctly? I didn't want to screw that up.
You pronounce it perfectly. Even my mom would recognize it.
Okay. So I'm guessing when you talk about, I want to start with the basics. When you're talking about your research on donor-derived infections, I'm guessing cytomegalovirus. What else? What else are we looking at that's donor-derived?
Also, kind of strange things like worms,
malaria,
like strongyloides, things that we use ivermectin for. Malaria. All these Chagas disease. So I think a lot of times, when we have donors that donate organs, their immune system is intact. So many chronic diseases they might have been exposed to during childhood that are kept in check. When we suppress the immune system in the recipient after the organs have been transplanted, they kind of come alive, almost like Walking Dead. So it's our job to kind of adjudicate the donor, make sure it's safe. When it gets in the recipient, we make sure that we institute meds, additional therapeutics if we need to kind of anticipate the bad stuff that can happen.
How do you deal with Chagas? I mean, that's not something you typically see in this country.
Actually, it's more common than you think because,
although I would say, given how Hollywood made it seemed like anybody could get a tsetse fly bite and develop sleeping sickness. But the Chagas is transmitted other ways as well, obviously this way. But go ahead.
Yeah, through blood transfusions. So we started, in the U.S. we started screening blood products for Chagas disease, and in transplantation it was really recognized early because when people might have been exposed to Chagas in Central South America, come to this country, or even if they weren't there and they were exposed to it by birth through maternal transmission, again, they die in a car accident. They fortunately donate organs to someone, but then that Chagas can come alive in the recipient.
So crazy. And strongyloides, for people that don't know, is a worm that comes in through your feet. Again, these are all essentially tropical illnesses, things we don't see here that much. But in terms of things that are here, does leptospira and Borrelia and I'm imagining tularemia and all, everything gets in, I guess, this way.
Yeah, everybody can get lots of things, but I think of the things that we most worry about in the US from US transplantation in terms of things that recipients can get, the big three are HIV, hepatitis B, and hepatitis C. But we've done a lot of good molecular screening, moved away from antibody testing to looking, using PCRs to look for fragments of these viruses in donors so that it makes it safer for recipients. So those are examples of things that we've
really
done a lot with. I mean, in California, we worry about cocci from, valley fever. A lot of times people don't know they have it in them, but then we see it in the recipient. West Nile virus, those are things that we've seen recently.
And not CMV, huh? I always think of that as the one that is so, or is that
CMV is
axiomatic?
80% of people have been exposed to CMV. So it is something. The reason why I guess I hadn't mentioned it because it's so, I think, well worked out and well honed out, and we have good therapy in terms of preventing people from having it rear up later on. But it is still very challenging. Didn't mean to minimize it.
The only reason I bring it up, it's on the MKSAP board reviews for infectious disease. They make a lot of CMV, but not every year. It's an every 3-year thing. It's weird what they decide to put in the MKSAP. It just depends on who the contributors are. And I remember at least 2 out of the last 9 years, CMV was a big deal. So I assumed it was something you guys were struggling with all the time.
No, definitely. I mean, yeah, somebody must like CMV who writes those questions. True.
Right, exactly. Just to make the boards more challenging. So let's talk a little bit of COVID here and vaccine therapies and whatnot. I want to get to vaccine, but before I do you have any opinion about some of the antivirals that are coming our way, the molnupiravir and some of the other products that may yet hit market?
I am so excited about molnupiravir. First of all, who wouldn't be excited about a medicine that's named after Thor? First, it is kind of a real great inspiration, but nevertheless, it's the first oral agent that's an antiviral that I think is so hopeful. And although the results that we've been seeing and for which they've submitted FDA approval for is really in the realm of early treatment, similar to monoclonal antibodies, but the real benefit will also be in post-exposure prophylaxis. So somebody in nursing home has COVID, the other people around, they don't have COVID yet. You give them this pill, we do with influenza and Tamiflu, and it's likely that it would not progress to COVID. Household, similar thing with post-exposure prophylaxis, easily available, probably from a drugstore. You just run out and get it. And at least in the trial so far with early treatment, prevents about 50% of people from getting into the hospital within treatment of 5 days, similar to what we do with Tamiflu, and relatively well tolerated. In fact, people who were on placebo, more people discontinue drug on placebo than who actually got molnupiravir.
Interesting. And do you worry about medication resistance? Are you worried we're going to need to put combo therapies together? I'm curious. I've not seen much about that. It sort of struck me as curious that people aren't worrying about that.
Yes, I mean, interestingly, with influenza and Tamiflu, we haven't seen too much resistance, but I think that is always something in the back of our mind, particularly with when you think about a virus, and if you think about the history and story of HIV, you put somebody in one thing for treatment, and then over time it develops resistance. So, it's something that's definitely going to be watched out for. And again, molnupiravir is the first gen kind of drug. Roche and Pfizer working on other oral agents too. And you're right, in the future forward, we can imagine combination therapy that people might institute at some point as an oral option.
And I guess it's not, these are not prolonged therapeutics the way it is for HIV. So there's not so much time for the virus to develop a resistance pattern. So that may be why it's not a prominent in people's thinking right now. The other thing, a question I had is I want you to address a sort of rumor that's flying around out there, which is that vaccines can accelerate drug resistance through putting evolutionary pressures on the virus. Address why that does not happen. And by the way, he embedded, it seems to me, in that story is this notion that the present mRNA therapies are therapeutic rather than vaccine. I think of vaccine as anything that prophylactically activates the immune system to prevent an infection. That's a vaccine therapy. But what about the evolutionary pressures caused by vaccine versus therapeutics?
Well, first of all, just the idea of a vaccine being preventative, as you said, there's nothing there. So like the traditional way we think about resistance on an individual probably won't happen because, again, there's no virus mutating because you're preventing the virus from getting there in the first place. From a population perspective, which is what you're getting to, could it be that vaccines will somehow lead to the virus mutating in a way that it's going to evade the vaccine? I don't think that's going to be true. So far, if you were a virus and you had a superpower that you could hold on to, and so far viruses, SARS-CoV-2 seems to hang on to one superpower, and the superpowers you have in your menu include, are you going to be more transmissible, be more vaccine resistant, or are you going to cause people to get sicker? It turns out that the one that survives the most, the one that seems to be viruses or SARS-CoV-2 variants that prize transmissibility. The one vaccine resistance and the ones that we know about so far are like beta in South Africa or gamma in Brazil. They tend to not proliferate as much as Delta has overtaken our landscape. So, I don't think that the vaccine resistant types will emerge, and I don't think that vaccines would lead to the rise of the mutants, so to speak.
Yeah, so that doesn't keep you up at night so much. That's what I worry about, is some sort of, I don't believe the argument there's increased evolutionary pressures on the virus for the very reason the way you set it up, which is there's no virus there getting pressured. That's not how it works. It's when it's under pressure is when it evolves in a certain direction, and that's therapeutics. I do worry about that. But then again, it's not about the infectivity. It's about surviving the therapeutic. It's about getting around the therapeutic, which is a superpower, right? I mean, that's what infective agents have as a superpower, another one, or a sub-superpower. So, and there's a couple of sub-deltas out there that are maybe a little more infectious. Why don't those take over?
That's an interesting question, but I also think it's something we don't fully know yet. I mean, many people have blamed the rise of the recent surge in the UK based on a sublineage of Delta. We've heard about Delta Plus before, but it's like one of those Delta Pluses. And, it could be that that's just the strain that's prevalent in a particular region and doesn't have extra transmissibility or vaccine resistance. But as we increase and open our borders more to international travel, including people from the UK, where this sublineage is more common, it will be interesting to see whether or not it takes over the landscape. But the good news for these variants of Delta so far is the parent is Delta. It's still vaccine sensitive. It's still monoclonal antibody sensitive. Even if it takes over, it's really predominantly in people who are still more susceptible, meaning they don't have an immunity either from natural infection or from vaccines. And it's not really going to take over the body of somebody who's been vaccinated, so to speak.
Excellent. So I wanted to ask some specific vaccine type questions. I'm starting to wonder, much like the HPV vaccine, if we shouldn't be thinking about the Pfizer vaccine as a 3-vaccine series. 1, 2, 6 months later, 3. That's a vaccine series. That's not a booster. Is that what's likely to happen with Pfizer? Or is it going to be an ongoing booster relationship with the host?
So like every infectious disease doctor, my answer is it depends. But I think the first possibility, what you mentioned, is my prediction. I think there are many, many vaccines, as you pointed out, Drew, that are given in 3s, measles, mumps, and rubella, human papillomavirus, and hepatitis B vaccines. And if you look back retrospectively at what happened with Pfizer and Moderna, they were really concentrating on the 1-2 hit within a month. And if you look at some of the other vaccines we normally give, you kind of have to give the immune system a little bit rest and then remind it several months later. And that boost is really what takes us across the finish line many years, more than 6 months is what people are worried about. Maybe the boost will only last 6 months, but personally, looking at the experience of other vaccines, it probably will last years. But the caveat is, if there is some super variant that's vaccine resistant, or appreciably looking so different that the vaccine antibodies don't recognize them, we may need a reminder, not for our immune system from the original variant, but because it looks so different. But I think overall, I think that the 1-2, wait several months, number 3 will probably last much longer than just 6 months, or it's not going to be an every year thing unless we have a scary new variant.
Got it. So what about natural immunity? I'm finding the landscape a little confusing with that. We have gone from ignoring it to saying it's 27 times greater to saying the vaccine is 5 times greater. It's all over the place, and they look like stupid headlines to me that make almost no sense. I'm guessing, well, I've done a sort of a full antibody, not really a widely commercially available antibody profile myself. So I'm an N of 1. I'm an anecdote. And as an N of 1, I have had sustained nuclear capsid antibodies, everything. My antibodies have been way up, including neutralizing antibodies have remained way up, which has been very reassuring. I couldn't move about without getting the vaccine, so I got the Johnson & Johnson vaccine just so I could move around and show my vaccine, card. But I didn't think I needed it. And it did boost, particularly, of course, my spike protein. And some of my neutralizing antibodies went up a little bit too, which was interesting. And just to flesh my story out, people have heard me tell this story, which is I woke up day 2 after the Johnson & Johnson vaccine with a spontaneous raccoon's eye on the right, which is the presenting feature of transverse sinus thrombosis, which freaked me the hell out.
You had a black eye.
I had no headache. I had no vision problems. I had just a spontaneous raccoon's eye. I thought, looking in the mirror going, really? I'm going to be the only male with the transverse sinus thrombosis? That's what's going to happen here?
No, I had to go put the dog in the cage. Sorry, Drew.
No, that's fine.
But
that's fine.
Did you guys talk about switching vaccines?
We're getting into. That's what I'm heading towards. So with that background, natural immunity, weird reaction to J&J, before I ask you, should I switch vaccines, what about taking a second Johnson & Johnson vaccine and completing that vaccine series? Should I do that? Or is it an unnecessary medical procedure?
That's a great question, Dr. Drew. I think to answer that question, let's go back and first talk about natural immunity by itself. I love natural immunity. You ask any infectious disease doctor, they love the idea that many times when you get a pathogen naturally, you get a really robust immune system. The problem is this is a shapeshifter of a virus, and we don't know what the enemy coming up next is. And it turns out that natural immunity by itself in the history of COVID hasn't been that great in the long hauls. In other words, the people in Manaus in Brazil, they got hit hard first, and then they got hit hard again by the Brazilian variant, which is Gamma. So that's like the natural experiment. And then we have the state of Kentucky, where people were cataloged who got infected in 2020. And lo and behold, they got cataloged again in 2021. They had the same people, everybody in database. If you go vaccinated, you were twice less likely to get Delta as somebody who was unvaccinated. So first of all, you could get reinfected if you were infected naturally, and if you were gotten a vaccine based on the new variant. But it turns out you were much less likely to get reinfected, at least from that data, if you were vaccinated.
Well, let's hang on.
And then last thing.
Wait, hold on. Before you keep going to the last thing, because that may be an expression of the variance of the antibody response, in that my understanding, it clusters into 3 groups, right? Rapid fall off in antibody, mildly, 6 months drop off, and sustained antibody response. So the reinfection might have just been in the lower 60% group, right?
Correct. But we can't really predict who is going to get, who has, unless you do all of the.
Well, we could develop technologies that could do that, right? But we've just said vaccine everybody, as opposed. So I've been watching myself, and I'm trying to be a little more sophisticated about it and take the vaccine when it's warranted. But we could do it. We just haven't yet standardized how to identify immunity, right?
Correct. So therein lies the difference between like a public health population intervention and an individual intervention, which I think we have the technology for. We can like phenotype your T cells. I can tell you what your T cell response is.
Yeah.
I can like biopsy your bone marrow and your lymph nodes and tell you what immune cells are there. You wouldn't like it. You probably wouldn't like it.
No.
You would like hate it more than your raccoon size. But I mean, it is something that I can do.
No, I totally understand. That's a very funny response.
I just think you should take the Pfizer.
Well, all right. So you were gonna. Yeah, I interrupted your talk about natural immunity. You said lastly, you were about to complete that thought.
Oh yeah. Lastly, I think when you look at the biology, it is actually quite fascinating because people who got infected naturally have a very long-lasting immunity when you look, more than 12 months so far in these various tissues I talked about. The holy grail actually is neither vaccine, probably natural immunity, it's the hybrid immunity, which you have. So I think those responses, which is called hybrid immunity, vaccine plus natural, ends up giving you the best of all because, we look at all these antibodies, but, the T cell repertoire is probably going to be much greater with hybrid immunity. It's just hard to figure out on a policy level how we can determine this person has that, this person has that, they don't need it. But for you personally, you're probably okay. But, to give you the benefit of doubt, say you didn't have COVID before, I would follow up with J&J probably with an mRNA vaccine. Given, we can talk about the NIH study, even though there's not clinical outcomes. But I think the robust antibody response in that and the easy availability of mRNA vaccines as a booster would probably be my recommendation, just like Sue said.
Okay. All right. I want to dig into that a little bit deeper. But first, I want to talk about hybrid immunity. And that is quietly under people's breath. And I've even saw it on the CDC website for about 5 minutes and they took it down. There's sort of well, we're not really saying this out loud, but really we think everybody eventually is going to have hybrid immunity. Is that true? Is that really that's the long ball? That's the long game, right? Which is get mild illness, treat it with molnupiravir. Well, get mild illness because you're vaccinated, get the hybrid immunity and get a short illness because you treat it with molnupiravir. That's what the world's going to look like in a year or two. No?
I think so. I mean, some pundits say that COVID is going to march around the world until it's touched everyone at least once. I mean, it may look very different, like you pointed out. That's a very benign look that I think people will be fine. You wake up, you're vaccinated, you have a cold, you go to the pharmacy, you get your molnupiravir that you have prescribed, you take it within 5 days, you have reduced symptoms, you can go back to work. And then you have the superpower of hybrid immunity that will probably take you on through, and then COVID will go away in the world. Well, it will be minimized. Probably won't go away.
Yeah, minimized. Right. That's what I think. I think that's what we're going to have to aim for. I really do. And the fact this world where it's zero COVID is just like an insanely maladaptive, inappropriate goal. It should be hybrid immunity, mild illness. Let's get on with this.
Yeah, I mean, the experiments of Australia, New Zealand, even Singapore, I mean, they all failed. And it's really surprising to me that China is still trying to go for zero COVID. I think it will fail, but, we can see what will happen.
But my question really was, you've confirmed my opinion, but my question really was, are people saying this out loud in important circles where people are thinking about these things?
People are starting to talk about it. I think the reason why it's been quiet and hush-hush is because people don't want to put all the eggs in the natural immunity basket. And I think there's a fear that by exposing the power of natural immunity, that it may deter people from getting vaccinations. But like we was both saying, it's the combination of both that probably is the holy grail. And I think personally, vaccines is more reliable, standardized. So I'd probably get a vaccine first for sure. And I wouldn't like risk getting natural COVID first. That's all.
And the hybrid immunity of vaccine then infection is the same as the hybrid immunity from infection then vaccine?
Probably. But there's a riskier enterprise by getting the natural infection first.
No.
Long COVID, all of that
Of course, clearly. And everybody, if you're going to do the risky way, have the monoclonal antibodies and steroids ready to hand. I think the steroid story, I don't know how you feel about this, but I think the steroid story is more powerful than we really have figured out yet. Because when people that do well, early on in the infection, I inadvertently treated somebody with steroids thinking she was having just an exacerbation of COPD. And she did really well and really should not have, because she was all set up for bad trouble. So I've been watching that, and I think that's more of a thing than we yet know. You agree?
Yeah, totally. I mean, if you look at, there are some other sort of anecdotes or data that's interesting to indirectly. So in my own peeps, the immunocompromised patients, we expected them to do really, really poorly with COVID, but actually it didn't. And because a lot of them are already on anti-inflammatories, and the same is with people with some rheumatologic diseases, they ended up not doing as bad as we thought, because probably the anti-inflammatories like steroids modulated their response. Because if you remember, of course, there's the viral phase, the virus making more of itself, and then the body gets angry by making the inflammation, and it's the inflammation and the immune response that kills you. So if you're like set up shop where your immune system is meant to be very sluggish, it sure increases your susceptibility to get infected. But if your body doesn't get ever super angry to really cause you to do poorly and go to the ICU and all of that stuff.
Yeah. I've seen more enthusiasm for the JK inhibitors lately. Should we be using those early?
I think it's hard to say. I mean, I think some of the other anti-inflammatory drugs like tocilizumab and baricitinib, they haven't really panned out in the data as well, and they only have panned out really for sub segments of people. So in other words, somebody who's about to go to the ICU, but they're not really on a vent, you might think about one of them. Somebody early mechanical ventilation might get another one. But you're right, but who knows if they would have done better in earlier disease. But so far the studies haven't looked at that population hasn't been as huge in the early disease part for those other anti-inflammatories. But steroids are cheap. They're like $7 a pop, versus $1,000 to $2,000.
Yeah. I am, though, hearing some more enthusiasm for baricitinib. I mean, tocilizumab is already in clinical pathways in some hospitals, but baricitinib keeps coming up in people when I talk to people, but may or may not be real. Who knows? Kindergarten, they're saying, Sean is saying, is JK Junior Kindergarten. No, it's the cytokine storm inhibitor. It's the baricitinib.
We get censored this week. If you're on YouTube, we'll see you over on Rumble. We're using a lot of big words here today, but
well, now that we're not using
I know. I was surprised how many big words we're using.
Well, we're not using the forbidden words. I mean, we can't. I was talking to a guy from Cleveland Clinic last week, and he mentioned the use of hydroxychloroquine early before anybody knew what was going on. And I had to say, I said, were we allowed to say that now? The doctors were doing that? Can we say that out loud without being canceled, for God's sakes? And I guess now we can say that. We still can't use you use the word to treat strongyloides. We're not allowed to say that word on YouTube. Do you know that?
Oh, no, I didn't. But we can use it.
You're not allowed to say. It's an anti-helminth that every refugee from a foreign country who comes to this country has to take. The CDC has it on their website. They must take it for 5 days after entry into the country if you're a refugee. But you're not allowed to say it on YouTube.
What if I was giving a lecture on strongyloides?
I know. Well, you can't do it on YouTube. That's the insanity. The AI will kick you out, and then when you try to make an appeal, they're idiots and don't understand what you're asking them, and so you just go, just don't do it again. Is sort of their response. It's really bad. It's really nasty. I've had. Go ahead.
Well, I was just going to say, what if I wanted to treat drug-resistant lice or Norwegian scabies? Then I would also be limited.
You can't say the word. What if I want to treat lupus with the HC drug? Well, you can say hydroxychloroquine now. That one all of a sudden you can say again. Yeah, because we said it with the...
Not here.
All right, well, not the two together in any event.
Now.
But if I want to say, I want to keep my lupus patient on hydroxychloroquine during pregnancy because it's so inert and safe, now I can say that. But 6 months ago, I couldn't say that. This is the insanity we're into these days. Anyway, I want to get to the switching from J&J to mRNA vaccine. There was some pretty good data on J&J and as showing very powerful response in the naturally immune patient. I felt pretty good that I took J&J. But in spite of that data, and that was after the one vaccine, but in spite of that data, you still say switch over to mRNA. Why such enthusiasm to switch from J&J to mRNA?
Well, I wouldn't say that I'm wildly enthusiastic, but if I had a choice, and I'm mainly, again, thinking about maximizing the immune response. First of all, Team J&J has always had been neglected a little bit, mainly because we didn't have enough, population to have robust data to make some recommendations about. And I think they were always number 3 in the medals count. It was an outreach vaccine originally conceptualized. Many people thought J&J should have been a 2-dose vaccine, just like AstraZeneca, and the other adenovirus vector. Yeah. So now it's back to 2. And then, I think that, that's kind of where we're coming from. There have been a little bit more issues with clots with J&J. But again, that's not limiting my enthusiasm. And I think with all things being equal, I think what's swinging many people into the mRNA camp is mainly the small study, although small and not clinical outcomes, done by the NIH, where they looked at 9 groups of people. And if you had J&J, Pfizer, Moderna, you got one of the other 2 as a mix and match strategy. Turns out when you had J&J followed by J&J, you had a response that was like 4 times. If you had J&J followed by Pfizer, it's like 32 times immune response several weeks later. If you had J&J followed by Moderna, you had like 76 times response. Sure, it's not like, fewer people going to the hospital or having symptomatic infection, but it was really kind of where things were going. At the end of the day, I'm not really saying use this or use that, but maybe the mRNA vaccines are definitely more common. You call up any Walgreens, you probably have to go hunting to find one that has J&J. Like a zillion of them will have either Pfizer or Moderna. So that's what I'm saying. If it's convenient, it's flexible, it's safe, and I wouldn't necessarily rush to get a J&J just because I had a J&J.
I like the idea of...
Also, there are good things with J&J too.
Such as?
There are good things about J&J. There have been this small study that people don't really talk about a lot, but when you look at, although it's like the bronze medalist in the Olympics of vaccines and Operation Warp Speed, when you look over 6 months, when you do get an immune response, it doesn't really seem to waver over the 6 months versus Pfizer. Moderna also a little bit less steep than Pfizer in terms of decline, but J&J is like swimming along at 6 months, which is kind of interesting, I think. And the reason why, I think when he did get protection from J&J, it was a little bit more durable. And that was kind of an interesting feel. And also it's a gift that keeps on giving. So the more you check antibody levels after a second shot in the J&J booster studies, the antibody response keeps on getting better. So it's like fine wine. You check it at 4 weeks, 2 weeks is this, then it's several-fold higher at 8 weeks, and then it just keeps on going up.
I'm inclined to get another J&J. I got to tell you, I am. What do we think the mechanism is of the clotting? It's the PF4, right? system?
Yeah, so it's a PF4. It's similar. Yeah, it's basically. Yeah, I'm not that expert in clotting associated with J&J and that particular adenovirus vector vaccine, but it basically stimulates something similar to clotting feature we see in people who get seriously ill with sepsis or infection in blood, where you have this clotting and unclotting and using up of factors, but then you should have thinner blood, but then you overclot. So that's essentially the path of Fizz.
Do you think I've been thinking about that? That's a good way of describing it, by the way. And I've been thinking about it, and I've been trying to decide, is aspirin therapy an appropriate thing going if somebody's worried about J&J reaction? Because bleeding is one of the problems with J&J as well. So what do you think?
I think if you are eligible to take aspirin for prophylaxis, I don't think it will hurt, but not like a therapeutic dose of aspirin for therapeutic blood thinning or anticoagulation. But say you're eligible to take an aspirin, why not? If it makes you take
like
the aspirin for other reasons, a baby aspirin, 81 milligrams, then yeah.
So,
but again, there's no data around that.
Where are you in children? I know we just today, the FDA approved, or at least provisionally approved, 5 to 12. What are your thoughts?
I think I support giving kids 5 to 11 vaccines for a few reasons.
Mandating?
The first is, sorry?
Mandating?
No, well, I think that eventually, with enough notice, not mandating it now, I think it has to have full FDA approval. I think we have to have enough experience in the population. And then I think a mandate in the future would not be out of realm. It's kind of early to say for sure what the experience in the population would be. I'm very optimistic. It's 1/3 the dose. We already see fewer chills and fewer fevers in the trial of 2200, although small, compared to the full dose in older adolescents and young adults. So I'm hopeful that we won't see any untoward effects. Most of the bad stuff with vaccines, when you look back at development of kid vaccines, happened in the first 2 months, and we haven't seen anything here yet so far. So, I'm holding on hope, and I don't think that I'll be wrong in this situation.
Discuss, if you can, people who are nervous about how drug companies report their data for vaccine research and therapies, as well as how somebody like you and I look at things like the VAERS system and other reporting system once there's widespread distribution of a vaccine. Because I will just say that non-clinical people don't understand what we see. When there's trouble, we see it. It's obvious. We start seeing it right away. It comes in. But help people understand if they're worried about, first of all, the drug company distorting data or something, the VAERS system underreporting or not being watched or not being made prominent, and what it means to, why physicians know when things are in trouble. Those are sort of 3 topics. Go ahead, see if you can make something of that.
Yeah, that's a complex 3-part question, Drew. I'd probably be sort of like puzzling over that on the exam if I had to. But let's take the first one. Drug trials. They are, they're very, very, very controlled. Control is the second letter in a 3-word randomized controlled trial, RCT. So control also means that people are like doting over these people in trials. So when you get a side effect, you have to describe it as an investigator as it likely, unlikely, and then you put all those things down. There's a category of serious adverse effect that would probably drive someone to the hospital. So these are all cataloged. But the problem with drug trials in general is that, maybe it's inherently biased because a company is running the ship. But again, the company wants something that will work out for them in the long haul, which is to try to be as responsible as possible. Maybe I'm being naive in that sense. But again, if you want to maximize profits, you don't want to fudge data because it's not good for the long haul. It's maybe good for the first 6 months before people figure you out. But again, you probably want to do things right That's how we approved lots of drugs. The second thing is, once it's out, then people start using it in real life and they report it to VAERS. And I think the story of J&J is probably a good illustration of what happened. So, so people start getting things. So this thing, blood clot in the cavernous venous sinus system, or CVS or CSV, depending on how you say it, people start noticing that. It wasn't in the trials. So what they did in the US was they saw it in Europe coming. It was kind of like the storm coming over with AstraZeneca. And then with J&J, we started seeing that in younger women. So what they did was, first of all, they went to the VAERS system. There might have been a signal there. And what they did was pause. So pausing does a few things. We think pausing means people don't run out and get it, but it also alerts clinicians to say, retrospectively, look at the patients who they've seen and maybe say, hey, I saw that person with a clot. Did they get a vaccine then? Because I might have thought the clot was due to something else. So that pausing allows that to happen. And then they go back to the data and refine it. But, it is as good as it could be, but it's not a precise science because lots of people can get things in real life and coincident with getting the vaccine. As it's hard to necessarily ascribe causation. So then you look at other things like, what's the risk benefit and all these kinds of things. You can get more clotting, several-fold higher with COVID than with the vaccine. And these are all things that go into the final population level recommendation, which may be different from an individual patient in front of you.
Yeah, that's I think is the point, is that the practitioners start to see this stuff, and then it gets correlated with population data, and that's how it's almost uncanny to me how quickly information passes in our profession. Very quickly people are alerting each other what's going on. I had never heard of something called cytokine storm, and 5 days into the sort of this thing hitting the country, I was discussing that with my peers, or something we were calling cytokine storm. And in that 5 days, how we look at inflammation in an ICU setting changed completely right then, boom. And something we learned about this illness was shared throughout the country, which was nice. But at the same time, some super uncanny things happened, such as we kind of froze in place and became afraid to think for ourselves. I want to talk to you about that. But more importantly, I want to talk about something that's happening now, is why isn't every physician aware of the monoclonal antibody therapeutics? It's so bizarre to me that it's just uncanny to me that people aren't aware of that. But I have to take a quick break, and we'll come back and we'll discuss those topics. I see you shaking your head and you're as incredulous as I am, but I hope you have some hypothesis as to why this happened. It's really kind of weird to me. I received monoclonal antibodies last December. We're talking a year ago, and it kept me out of the hospital. One of the things that I immediately did was went out on social media trying to educate people, because I was shocked that the public health officials weren't educating the public about how to stay out of the hospital. They were, stay home. No, if you get sick, find a physician, use telemedicine, get the monoclonal antibodies, stay out of the hospital, talk to your doctor about steroids. There's a lot we can do. They did none of that ever. And to me, this is all an astonishing part of the story of the COVID pandemic. So we'll talk about that. I'll get your thoughts on it in just about 3 minutes. Be right back. I want to give a shout out to our good friends at Blue Mics. If you've heard my voice on this show anytime over the past year, including right now, you've been listening to Blue microphones. And let me tell you, after more than 30 years in broadcasting, I don't think I have ever sounded better. But you don't need to be a pro or have a fancy studio to benefit from a quality mic. You may not realize it, but if you've been working from home or using Zoom to chat with friends, you probably spend a lot of time in front of a microphone. So why not sound your best? Whether you're doing video conferencing, podcasting, recording music, or hosting a talk show, Blue has you covered. From the USB series that plugs right into your computer to XLR professional mics like the Mouse or the Blueberry we use in the studio right now. Bottom line, there's a Blue microphone to fit your budget and need. I can't say enough about Blue mics, and once you try one, you will never go back. Trust me. To take your audio to the next level, go to drdrew.com/blue. That is drdrew.com/BLUE. Anyone who's watched me over the years knows that I'm obsessed with Hydralyte. In my opinion, the best oral rehydration product on the market. I literally use it every day. My family uses it. When I had COVID, I'm telling you, Hydralyte contributed to my recovery, kept me hydrated. Now, with things finally reopening back around the country, the potential exposure to the common cold is always around. And like always, Hydralyte has got your back. Hydralyte Plus Immunity, my new favorite, starts with their fast-absorbing electrolytes and adds a host of great ingredients. Plus, each single serving easy pour drink mix contains 1,000 milligrams of vitamin C, 300 milligrams of elderberry extract. Hydralyte Plus Immunity comes in convenient, easy to pour sticks that rapidly dissolve in water, make a great tasting drink, has 75% less sugar than your typical sports drink. It uses all natural flavors, gluten-free, dairy-free, caffeine-free, non-GMO, and even vegan. Hydralyte Plus Immunity is also now available in ready-to-drink bottles at the Walmart next to the pharmacy, or as always, you can find it by visiting hydralyte.com/Dr. Drew. Again, that is hydralyte.com/DRDREW. Be sure to use the code Dr. Drew 25 for a special discount. Here with my daughter Paulina to share an exciting new project. Over the years, we've talked to a ton of young people about what they really want to know about relationships.
It's difficult to know who you are and what you want, especially as a teenager. And not everyone has access to an expert in their house like I did. Of course, it wasn't like I was always that receptive to that advice.
Right, no kidding.
But now we have written the book on consent. It is called It Doesn't Have to Be Awkward, and it explores relationships, romantic relationships, and sex.
It's a great guide for teens, parents, and educators to go beyond the talk and have honest and meaningful conversations. It Doesn't Have to Be Awkward will be on sale September 21st.
You can order your book anywhere books are sold. Amazon, Barnes & Noble, Target, and of course, your independent local bookstore. Links are available on drdrew.com.
So pre-ordering the book will help people, well, raise awareness, obviously, and it'll get that conversation going early so more people can notice this and spread the word of positivity about healthy relationships. So if you can, we would love your support by pre-ordering now.
Totally. And as we said before, this is a book that both teenagers and their parents should read.
Read the book, have the conversation. It Doesn't Have to Be Awkward, on sale September 21st. We are here with Dr. Peter Chin Hong. He is a medical educator at UC San Francisco Medical School, where he is a dean as well. And we were discussing before the break the incredulity that he and I both have at the fact that our peers seem to be unknowledgeable about the one of the and steroids too, to some extent. They seem reluctant to use that, but certainly the MABs. I'm shocked. What is your understanding of how that happened and why that is the case?
Well, when you were mentioning or introducing the topic, the only thing that was popping into my head was demonic possession, because that's the only reason why somebody shouldn't know about this medicine. It is, like you said, it's like one of the truly evidence-based therapies that we have for early disease. There's actually more evidence for monoclonal antibodies, and the evidence is better than for things like remdesivir, where there's no, the p-values are only in a subgroup of populations, and you put a lot of stock in remdesivir. So, I think like steroids, there's more, there's a lot of statistically significant benefit, and we haven't used it. It's free. The government spent more than $3 billion pre-buying a lot of monoclonal antibodies. It's effective against Delta, and we've only used about 50% of the supply, which is weird considering that 700,000 people have died in this country from COVID and could have benefited from monoclonal antibodies. If you look at the history, we've had an emergency use authorization since late, last year for one of the products, and then two more subsequently this year. So, it is mystifying. I think when I step back and I think about, as an educator, why this block is there, I can only think it's information overload, and then there are so many permutations and combinations. But again, of all the therapies we talked about, there are few that rise to the top of what is really evidence-based, and monoclonal antibodies is among them. So I'm not sure why it's not getting played, but I know, it may be resources, the siloing between outpatient and inpatient. I think it's easier to medicalize and treat a lot of people with things once you get into the hospital. But if you're on the outpatient, it's almost like the Wild West, or you kind of have to go through the desert, or you're Mad Max on a motorbike, and you're trying to find the monoclonal antibodies, because it's not easy to find and people don't know about it. So long diatribe,
and I think
to say that I'm not sure.
I get you. And I think it varies state by state. I talked to somebody in Florida this morning who was middle-aged, mild COVID, found it, got it, and they gave it to the wife for exposure prophylaxis. Really?
Yeah,
and I was like, amazing. This is fantastic. It's fantastic, yeah, right? That's Florida. That's Florida.
Well, Florida democratized getting monoclonal antibodies by having patients self-attest, whereas it's kind of locked in many other areas and states. So patients self-attest, and the other way they dealt with it in Florida is they had it at churches and theaters and community centers. Mm-hmm. Like the person called you can kind of go and get it. And it's not like they're not using evidence, but it is both for treatment of early disease and for post-exposure prophylaxis. And it's available because the government bought out a lot of it. So.
And people, the public thinks it's expensive and not for me. It's for special people. Wrong. It's for everybody. Your tax dollars already bought it, number one. And then the doctors, this is the more interesting part of the story for you and me, have been weirdly frozen in place since the beginning of the pandemic. And it's been the weirdest thing I've ever seen. And I think that some of it is that more of our peers are employees than we realize. And they're sort of stuck with whatever the system is telling them rather than allowing them to practice medicine, I suspect. Also, they became fearful of the social media and the mob, and being and getting their license encumbered for God knows what. I feel like there was a panic amongst physicians in addition to being encumbered by being an employee.
I mean, that could be it, because there is, again, there's been a lot of barriers to giving it in hospital systems. That's why it really has to be embraced either by another body, like a political body or public health body that's higher than the system, or the system has to believe in it. Mayo Clinic believed in it, so they were able to give, which is huge for a system. It doesn't sound like a lot, like something like 280 treatments a week. I mean, I think there's Oshner in Louisiana during the Louisiana surge was able to get up to over 1,000 patients a week, which sounds respectable. But again, if you think about your average academic medical center, you feel great when you're able to give 30 people a week. Because again, the outpatient versus inpatient silo is real. You kind of have to have nurses, you have to have people worried about infection, because even the Uber to get there, you can't get an Uber. Somebody has to bring you there because people are worried about getting infected. Because again, it's the difference between outpatient and inpatient. People have to observe you after getting the monoclonal antibody, which isn't necessarily a barrier. People have been trying to get around that by giving injections in drive-through clinics at homes. I think, but I think the case of Florida is, an instructive one, because they, again, made it for the people and kind of disseminated broadly, whereas otherwise it's kind of like a secret message and you kind of have to have the password to know about it.
Right. I got it from an infusion nurse in my home, and that's what was provided through Caremark at the time, which was great, which is absolutely standard care, and it should be the standard care, but God knows why it's not. But there's so many. Well, do you, as you look back on the pandemic, are there lessons to be learned that you sort of are thinking about?
Yes, I mean, I think there are a lot of lessons to be learned. I think one of the lessons that I learned was something you mentioned, Drew, which was that the way we as healthcare providers get information, I think early on in the pandemic, we were all learning together. So I think social media was very powerful then, by people sharing how we took care of patients, how we developed guidelines without somebody giving you a nice flow chart before. We were building the plane as we flew it. And then what happens is that's the clinical thing. And then there are all these therapeutics that get introduced in rapid succession throughout. And not only do we have to learn the therapeutics, we have to prioritize them, and then we have to readjust our schemas. So that and reaching out to a community was probably the first lesson that was very remarkable during this time. We couldn't have done it in 1918 when we had the influenza, Spanish flu, because we couldn't share information that way. So it was remarkable. The number two thing I think I probably learned, lessons learned, is that, I think, this has really been a political disease, and I think it's mystifying, but then one can say that was a naive view as a healthcare provider, where we use masks so often in the hospital for lots of things, and as an infectious disease doctor, we use masks a lot of times for TB and all these other reasons. So I think the politicization was kind of difficult for me to swallow, because I was just thinking health systems and healthcare and what's the evidence, and in this setting I want to use this, versus like thinking about what people thought it meant from a political perspective. So that was a little bit sad to me. And then number three was, again, a lot of times we have to and then this is kind of like the 2021 thing, looking globally, not because we necessarily have to think only about equity, but we can use patterns around the world to help predict what was coming. So Delta is a case in point. We saw Delta actually coming like a storm before it hit the US. And I think we didn't look outside with that clarity to see it go from India to the UK and then to the US. So we could have probably done a little bit more preparation and anticipation. Again, prevention, and then hindsight is always 2020. So those are just some of the lessons we learned. I mean, I learned that I really prioritize hugging, and I never thought I would miss hugging as much as I did. And I learned to make a lot of banana bread, but those are the silver linings from the pandemic.
And yeah, I mean, mRNA therapies and molnupiravir and lots of interesting Zoom and sort of technological advances in terms of how we care for patients through electronic media. But totally. you sort of hinted at the mask thing. To me, that we shouldn't let panic dictate public health policy. The fact that to me, the highest level of incompetence was public health officials not allowing people to lie down on the beach. You could go on the beach, but you couldn't put a towel down. That's disgusting incompetence, and it should be called out as such. It is frank incompetence, and that was a gigantic error, and they should call it out. And they should have been encouraging us to go out on the beach and going to the parks where this thing doesn't transmit. And you mentioned mask. What do we do with mask data? Do you make anything of the Danish study or the Bangladesh study?
I mean, I just, they are all, you can examine these studies from closely, and then kind of looking for flaws or not for flaws. Is it generalizable to where I'm at?
Like every study, though. We do that with every study.
Like 50%, did people wear the mask a ton, or do they kind of half wear it? But at the end of the day, the way I think about masks is the way I think about it in medicine. It's not like the be all and end all. It's not going to prevent you like a force field, but it is something that's easy to wear, and I'll slap it on if I feel like it. But I'm not going to put all my eggs in the mask basket. So that's the way that I overall feel about masks. It's kind of like the icing on the cake, but it's not the core way in which, because I have these other things now to protect me.
That's kind of the way I think of it. I think of it too, which is a Danish study. So neither the Bangladesh or the Danish studies really didn't show zero effect. It showed some effect. And if we want to have some effect, well, you can wear these things. But to pretend as though you're killing grandma if you don't wear a mask, well, that's insane. Now we're insane again. Now we're preventing people from going to the beach again. This is the back to the Panicsville, and that's the part I've objected to so much. This whole thing. And the other thing is I've learned about public health, the way we distribute public health authority. I mean, we gave it to a sociologist, and non-clinicians were making major decisions that had huge risk-reward concerns that were not taken into account at all. And now we have those concerns coming to bear, i.e. mental health consequences, et cetera. And that to me was just troubling, just deeply troubling.
I think what you're getting to, Drew, is the whole idea of how we might put science onto a policy landscape or into the society, which is, I was on Left, Right, and Center with Josh a few weeks ago, and I think he put it most eloquently when he said, scientists can give the science, but it's up to society to use values to translate that science into what people want to do. So I think, at the end of the day, speaking to your sociologist comment, that is what I think, that's how the dust settles.
I heard Vinay Prasad say almost that exact same phrase about science being, just being say it again. What was the phrase you used? The science having a value to it, or
science gives, it's a guide, but then society has values in which you can interpret the science and then put it into what society wants.
Yeah. The science needs to be interpreted within the context of the values of the culture, I guess, is what
somebody on YouTube named Charlie Pritt said, "I'm so embarrassed that I caved in and listened to my wife and left the mail outside for 2 hours before bringing it inside."
Well, that was during the panic, and people were, didn't know. We were watching YouTube videos on how to wash your, how to treat your produce like they were surgical instruments for use inside the body. That was crazy.
I never cleaned my groceries. I never did that.
No, we went to New Orleans, and they lifted the mask mandate, and then I got my nails done when I came back to Pasadena, and I had to go next door. I was going to pick up some cupcakes, and I forgot my mask. I forgot about the masking. Everybody had masks walking down the street in Pasadena
outdoors,
and I walked in, and she's like, "You have to put a mask on." I go, "I can't. My nails are wet. I can't do it." And I said, forget it. I don't want these stupid cupcakes. And I walked out. I was just like, when is this going to stop?
This will forever go down as the cupcake incident.
I know. I was just thinking whether or not you got cupcakes at the end of the day, because I probably would have.
No, I didn't need the calories.
Did you get the bun cake or not? That's really funny. Well, listen, it was a pleasure to talk with you. And I feel like I learned. Well, there was a lot of information you gave me at the beginning about infectious disease and dormant infection in donors. I didn't realize there was so much of that. I always thought of the big three, as you say, and CMV. But that's fascinating. That's an interesting way to see a lot of disease that you might not see in any other context in this state anyway. Although I used to see a lot of worms of all types, not strongyloides, because that really is African, right? But from Central and South America, a lot of different kind of worms, including Chinese liver flukes and
oh wow, that's neat.
All kinds of Ascaris, and I remember I saw something in a common bile duct. I can't remember what that worm was, but we use these antihelminthic medications like crazy. They were gigantic breakthroughs. They were a huge deal, and now you can't say the word on YouTube.
Dr. Peter, do you have a quick minute? Dr. Peter, this is Caleb in the control room. I actually have a quick question. So I have severe Crohn's disease. And so part of that, you had mentioned earlier about immunocompromised people, that you had noticed that strangely enough, they didn't seem to be getting COVID in higher numbers. Right as COVID was starting.
They didn't get Caleb, they didn't get the cytokine storm.
Oh, they didn't get the cytokine storm. Okay, that makes more sense, because I was confused. I've been very confused as to how I never caught COVID, because I was literally in the hospital in the end of February in 2020 when nobody really knew what was going on. They weren't doing any protection. I went into the hospital, severe symptoms. I spent almost like 8 or 9 days there. I went in there. They weren't doing anything by the time I came out. On Humira for 10 years.
And steroids.
On Humira and steroids. I did that. I had surgeries. Then I went back 6 months later, and I never caught COVID. And I feel like I would have been very highly likely. Have you noticed anything that could explain why? I was literally in the middle of Los Angeles as well.
I mean, there are two explanations. One is that you might have gotten it and not known it, because again, you were on these anti-inflammatories, so like your inflammatory system was kind of dampened. The second explanation is, and then the not knowing part is that they didn't test you. Or because in the beginning, nobody was, getting the test was like gold, you kind of had to be an NBA player or like Tom Hanks or something. to get it. But now, it's everywhere. And the second reason is, what we know from the second reason is just that you were lucky or killed. We know there's a good Swedish study where they looked like the entire population, Swedish and Scandinavia, they have like all these population databases. They looked at Crohn's in particular, and like what Dr. Drew was saying, the people with Crohn's tended to get infected more, like they went to hospital, but they didn't die more than the general population, which again suggests that even though your immune system is compromised because of the medicines you take, that bounces off with like not having that cytokine storm.
Right. Okay, that makes a lot of sense to me. I was shocked whenever I actually moved out of Los Angeles a few months ago, and I never caught it. I got tested for it many times, and I seemed like I was the perfect candidate to get it, of being right there at ground zero, Lucky, I guess.
I think it would be interesting for you to check your antibodies like Dr. Drew and see if you
antibodies.
Got it and didn't know it.
God
I should do that.
He gave you enough problems, Caleb.
Yeah, it's like he was done for the year.
I know.
Yeah, no more. Leave that guy alone.
He had a psoas muscle abscess that he was walking around with for 3 months that they finally identified. Yeah, it was bad.
I couldn't even lift my leg up all of a sudden, and it was bad. And that was after on Humira for 10 years and as immunocompromised as I could possibly be, and I somehow made it out without it, and then the vaccines came out, and
lucky with the psoas muscle, lucky with the COVID.
Exactly.
I'd rather have COVID.
There you go. All right. Well, thank you, Caleb. And I want to take a couple calls off Clubhouse, but first I want to let Dr. Chin Hong go.
Thank you so much.
We will follow you at PCH on Twitter. PCH, capital letters, PCH_SF, PCH_SF. You have to tell us what that means. What is Pacific Coast Highway, San Francisco?
Pacific Coast Highway, Peter Chin Hong. You can have lots of ways for PCH.
PCH. There you go.
That's his name.
I love it. I didn't even. I'm too.
And he's in San Francisco.
I got the San Francisco part. I didn't notice his name, which is foolish on my part. But so anyway, thank you for spending some time with us, and I hope if new stuff comes up, you'll be available to kind of help us make sense of it. We appreciate it.
And if we get censored on YouTube, we're also on Rumble, Facebook, and Twitter.
And you can always get me cupcakes too. Yeah, next time we can hopefully
I will bring you
cupcakes
next time.
And get me some cupcakes.
There you go.
That'd
be nice.
Fabulous.
That'd be great. If you're down here, let us know, please.
And we're going to show a picture of Drew's black eye pretty soon.
So is that right?
I found a picture on the Mike Corona show.
Fantastic. All right, let me take a couple calls here. Thank you, guys. Some people have been waiting for quite some time, and I want to try to get to them very, very quickly. We have a few more minutes for calls, and we appreciate you guys hanging out on Clubhouse with us, as well as those of you on the restream. I'm watching that carefully. I have clicked on somebody here who may have.
We had to weed out the weak. We want the real strong calls.
I wonder if this M. Homestead has left the building.
Okay, if you're on Clubhouse, wake up. We're over here. We're ready. We're ready for you.
Okay, I'm going to try another one that seems to have been around for a while.
I think we got demonetized on YouTube, so that's probably why we're not getting any more strikes.
Maybe. Hi, Jordan.
Hi there. Can you hear me all right?
We got you.
Okay, awesome. My question has to do with, I guess, natural immunity and the vaccines and this sort of hybrid immunity you and Dr. Peter were talking about. So I'm just wondering if you're familiar with the idea of original antigenic sin at all. It's also called like imprinting or the Hoskins effect.
Yeah, I looked it up once and did not spend a lot of time with it, but go ahead.
Okay, so I've just got a study in front of me here that's showing that for breakthrough infections, only about 26% of people are developing the anti-nucleocapsid antibodies, compared to about 82% of people who are unvaccinated. So I'm just wondering if there might be some effects on whether you get the vax first or infection first.
Right. So you're making a case that if you want to get hybrid immunity, you should get the infection first, so you get all those nucleocapsid responses as well.
It seems to me, yeah.
Yeah, he said he didn't know when I asked him that, because I kind of asked that question. He said, well, probably the same. But that's fascinating to me. I'm wondering how long after the natural infection were people being studied? Because I'm wondering if they had it and just it wore out, and some, then 60% are going to have a drop in their nucleocapsid antibodies.
For the breakthrough infections, it says it was within median of 30 days.
Okay, so the breakthrough, and those are the ones that had the lower nucleocapsid antibodies?
That's right, only 26%.
I don't have an opinion. I'm not sure he would have an opinion, but I appreciate you bringing that up. This is the kind of thing that we need to be thinking about. I don't know. Think about it. Let's say that's accurate. I still don't know that it's worth the risk of getting the natural infection first, right? If we're going to get hybrid infection anyway, I'm not sure if that's worth it. Starla, is that you? You suddenly came in here and kicked Jordan out. Starla, there you are.
Yeah, I'm not sure how I made it up here, but I do have a question. So I also have a couple of autoimmune issues, and right now I'm just treating it with prednisone. I've been exposed multiple times to COVID, and I've not actually come down with it. We've even done the nose swabs, and is there something with the prednisone? And even when I was taking some of the rheumatoid medications that may be preventing me.
So have you heard what Dr. Chin Hong was just saying to Caleb, who is somebody immunosuppressed with Crohn's? It doesn't appear to, in fact, they seem to maybe get infected at a little higher rate. They just don't get the complications at a higher rate, or it's similar to, or maybe less than what you would get if you were just somebody without immunosuppressants on board, which makes perfect sense. I mean, we're giving people immunosuppressants once they get sick to prevent the cytokine activation and the inflammatory
and plus
component
he's
of the
on
disease.
steroids all the time.
So he stays with the steroid, but he didn't get infected is the thing. And so Starla's asking, why not? Is it affecting my ability to get infected? And the answer is nobody has really studied that. We don't believe that to be so. We would but some people have innate resistance to the infection too. I mean, you could be one of those people, right?
Like me.
Right. Susan seems to be one of those people. But we are, at least from the standpoint of what PCH was just saying, what I've been thinking is it's really the complications that are reduced, not whether you get infected or not. And also not whether or not you have significant illness. It's really whether you get the really serious piece of the illness, which is the inflammatory piece. Okay, guys, I have to kind of wrap things up here. Time is up. We appreciate it very much. You have some very interesting things coming up. Dr. Antonio Damasio coming in here next Monday. That is a big deal.
Is it?
Oh, my goodness. He is one of the...
And by the way, Michelle Poe is killing it.
Yep.
She's the master booker of doctors. And she just said she thought she did a pretty good job today.
Yes, this was good today.
So I'm giving her a shout out.
And thanks, Caleb, for producing. Thank you, Michelle, for booking. But Dr. Damasio is probably the... he is the leader in neuroscience and psychiatry in thinking about things like consciousness and feeling and self. And he is really, we've had Dr. Alan Shore in here, who's one of the leaders in interpersonal neurobiology. Dr. Damasio is the international sort of superstar in this space.
Somebody just said, Dr. Kelly.
He wrote an important book called Descartes' Error. He also wrote The Feeling of What Happened, something like that, or The Feeling of the Self Comes to Mind. Self Comes to Mind, I think is his other one. And I think he wrote even another one called Brain on Fire or something. Maybe that was not him. But Descartes' Error is sort of his classic book. And he has a new one out where he has really reformulated the importance of feelings, so-called, what feelings are. And so we're going to get deep into the conversation about feelings, emotions, where they come from, how they're regulated, how they're perceived, and that relationship of that with consciousness. It's going to be a bit of a heady discussion, I suspect. But that'll be Monday at what time, everybody?
Next week?
Monday at what time?
No, Tuesday. Wait.
Monday at...
Wait, you're talking about next week. We don't get back till Monday. It's Tuesday, I think.
It's Monday. We have another guest on Tuesday I'll tell you about in a second.
Ooh.
So 4 p.m. Pacific.
Let's be hopeful that our
4 p.m. Pacific on Monday. And
all right. Hopefully our flight will make it back.
Right. And then Tuesday, let's see, we had somebody then too, did we not? Or is that because I'm leaving, we're not doing anything Tuesday?
No, Tuesday. We might have to move that guy a day if we have. Oh, you're leaving that day. But we're going to be here till Wednesday and then back on Monday, and Drew will be out of town for a week. So
okay, so we're doing something
so you guys can catch up on your last episodes.
Are you saying we're doing something tomorrow, right? That is with Dax
Holt and his buddy from their podcast that you were on.
Yes. Very fun guy.
I can't remember
for years.
his name though.
Used to see him on TMZ. He was the guy holding up the disgraceful pictures.
He was a nice guy at TMZ.
Yes, he was.
And then we have a lot of your mom's house fans here. I just want to say hi.
Great.
On YouTube.
Excellent.
Probably, I don't know. Do we have a little clip, Caleb?
I'm going to play that at the end. I have it lined up after the disclaimer.
It says, honor my degree. I don't know what that means. What does that mean?
Stay tuned for the latest episode on After Dark.
All right. I'm just looking at your comments here on the restream to see if there's anything else. I think we're not. There was a lot of call to get Robert Paul Champagne in here, which I suggested something like that over to the powers that be at your mom's house. I'm not sure that's going to happen.
Kind of their gig. So
that, and he's sort of used for the live shows. So I begged on some stuff there. Thank you to the folks.
They're afraid we'll leak out too much information.
Thank you for the guys over at Clubhouse. We'll be back there again. Probably
great calls at Clubhouse. Thanks, everybody.
Probably tomorrow. Thank you for hanging in there. And then everyone on all the other streaming sites, we appreciate you guys being here. And we will be back tomorrow at 4 o'clock, 4:15, something like that, Pacific, with Dax Holt, and then Monday with Dr. Antonio Damasio. We'll see you then. Ask Dr. Drew is produced by Caleb Nation and Susan Pinsky. As a reminder, the discussions here are not a substitute for medical care, diagnosis, or treatment. This show is intended for educational and informational purposes only. I am a licensed physician, but I am not a replacement for your personal doctor, and I am not practicing medicine here. Always remember that our understanding of medicine and science is constantly evolving. Though my opinion is based on the information that is available to me today, some of the contents of this show could be outdated in the future. Be sure to check with trusted resources in case any of the information has been updated since this was published. If you or someone you know is in immediate danger, don't call me, call 911. If you're feeling hopeless or suicidal, call the National Suicide Prevention Lifeline at 800-273-8255. You can find more of my recommended organizations and helpful resources at drdrew.com/help.
Man, after these voicemails, you got something that you're going to do for me that I've been trying to get out of you for a long time, but
I'm not going to suck your dick.
Now that...
It's not going to happen.